Hello and welcome to Nikolai's genetics lessons, and today I have prepared two very easy multiple choice questions for you. One is multiple choice question, another one is true-false question. As usual, I recommend you to pause video here, try to solve each problem on your own first, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. And here is the first question. In lilies, white flowers, that is W, are dominant to purple flowers, that is small w. If two plants that are heterozygous for flower color are mated, the offspring might have which genotype? And here is the 5 answers to choose from. And to give an answer to this question is very easy. Everything we have to do is just to build a Punnett square. So here we would have genotype of one parent, that is capital W and small w, capital W and small w on the other side, the genotype of the second parent. And when we build a Punnett square, we can see all the possible genotypes that these two parents can produce. So here we would have capital W, capital W, capital W and small w here, capital W and small w here, and small w here. As you see, here is a homozygous dominant genotype, here is heterozygous, and here is homozygous recessive. So all three answers A, B and C. So we have to choose answer D. All of the above would be correct answers A, B and C. And next, true-false statement. Proteins are composed of strings of nucleotides connected together by 5' and 3' and phosphodiester bonds. And this is exact description of the bonding in the double-stranded DNA. So as you see, here we have sugars that are connected by phosphorus, and each phosphorus has two bonds, and we call such bonds ester bonds, and because each phosphorus has two such bonds, we call this phosphodiester bonds. And this is covalent bonds, and that's why when we for example doing PCR, we can disconnect two strands of the DNA, because here we have very weak bonding. This is hydrogen bonding, but two strands of the DNA can separate and still would then anneal at the same sequence of the bases, would be conserved by the strong covalent bonding. And we can say that this is false statement. And proteins, and here is the structure of the amino acid. So nitrogen here, two hydrogens here, and two carbons here. On one of the carbons we have R group that would be different in each amino acid, and oxygen and OH here. Each amino acid has carboxyl group and amino group, and structure of each amino acid would be the same except for the R group that defines which amino acid this is going to be. And as you see, the structure of each amino acid, basic structure would be the same except R group. If R group would be the same as here, this is going to be the same amino acids, and amino acids are connected by peptide bond, connected by dehydration synthase. So one molecule would lose OH and another molecule would lose H. Together they would make H2O, or molecule of water, And as you see, between two amino acids we have strong covalent bonding, and if you would see changes in structure when you boil, for example, egg, this doesn't mean that this bonding would be destroyed. Actually, what cause this change would be change in three-dimensional structure. And as you know, three-dimensional structure of the proteins primary made by weak hydrogen bonding, but even if you boil an egg, the primary sequence of the amino acids would be the same, because here we have covalent bonding, just the same covalent bonding like we have between other atoms in this molecule. Once again, proteins are amino acids, and here we have example of the nucleic acids. Nucleic acids has 5' end and 3' end, and amino acids has amino end and carboxyl end. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.